We are talking about manufacturing this morning, which is my personal drum of the most important, most overlooked part of drug development. So really excited to have everybody here from Catalent. I'm gonna let you introduce yourself a little bit more properly. But just as a quick reminder, if you haven't signed up for the one-on-ones this afternoon, strongly recommend it. Great opportunity to just get a quick touch point and understand what capabilities are. Even if you have something that's a little bit weird, we were talking, come talk to me, because Catalent can do a lot of things other than sort of your classic small molecule, classic antibody, large molecule development. So with that, I want to turn it over.
I'm not sure that you go for the first.
Okay, relate then.
Where am
Why
I?
don't you start?
When I
You
advance.
probably want to be here-ish,
Okay.
because yesterday the person was standing over here and they were getting a little close to the camera.
It was working fine from here, so.
So is this my target here?
Yeah, showing up good.
Up or down, it was working earlier for me. There you go.
Okay.
Yep.
So it goes, okay, cool.
Yep.
All right, I just want to make sure. All right. Hi, good morning, everyone. Just wanted to say thank you once again to the TMC team for having us here and having us participate. We're super excited to be here. And thank you to all of you for joining us this morning, especially in person. I'm super excited that we can all be here in person. My name is Elena Valello. I'm an account director here at Catalent, and I support our early phase small molecule technologies. So my role is in business development and responsible for maintaining and building new and increasing business relationships. So I'm based in Maryland, and I've been with Catalent for about 5 years now. I'm going to start off just by sharing a high level of what Catalent does as a CDMO, kind of where we started, and then where we sit today. So I have about 5 slides to share, and then I'll hand it over to Brent. Do you want to introduce yourself since you have your slide here?
I know, what a weird place to put my slide. Yeah. Yeah. So guys, I'm Brent Moody. I'm a principal scientist in what Catalent lovingly refers to as the science and global science technology team. We serve as we report directly up to the chief science officer. We're a small group relative to the size of Catalent. And my boss says be a Sherpa, just make sure that client project technical needs are getting to the right place on the Catalent mountain. So I'm part of the North American tech support team, and we're usually engaged. What we're doing is we're usually sitting side by side with our business our representatives, just hearing out what the customer has. And for our later stage, we'll talk a little bit. A lot of my presentation talks about the difference between early development and late development. Later customers usually know what they need and they know where they're going, whereas our early stage customers do not. And so we provide that technology agnostic, business agnostic point of view and look at it. So we have a pretty diverse team here in North America and in Europe. I happen to come from an oral solid drug delivery background. So I'm 20, it says 22, but actually 25th year in industry. I guess that's pretty old. And almost all of that has been formulating small molecules for oral delivery. I do have some peptide and injectable background, and my first job in industry was actually an analytical chemist. So I get on my soapbox about having good methods in place if I'm not careful. And and that's my role now. I'm based in North Carolina, which is home to me. You hear me talking, you tell I'm not from California, but I came to Catalent by way of San Diego through a smaller version of Catalent, a small privately held company that was founded in 1999. I was employee number 12 in 2003. And we grew the business to about 225 people. We still do all the spray drying and the amyl sol dispersion work out in San Diego. So that's really where I have my path to get into Catalent. We were acquired in 2016, and I transitioned over to the science technology team. So now I work from a home office in North Carolina and usually travel around a bit. But this is the first time they've let me out of the house in 2 years. So I don't know. I have a.
I think our last trip was together before.
No, actually, well, you and I did, but I tell the story. It was the last week in February of 2020, and I was in New York City doing a lunch and learn at Alexandria with our DMPK guy, and I said, "Have you heard this thing about this coronavirus or something?" And he was like, "Oh, it's nothing. Don't worry about it." And I was like, because I had hand sanitizer. I was sanitizing my hands. It was kind of germaphobe anyway, and I remember him going, in Sweden, we know how to to be careful. 2 weeks later, he had a different tune. So hopefully we're past that. So nice to meet everyone. And I'll let you get back. You have 1, 2, or 3 slides, and then we'll jump on my presentations.
Okay. So just want to start off by talking a little bit about Catalent and sort of our history. So Catalent actually began back in 1933. We began as RP Shear. down in Tampa, Florida. So that site still sits within our network today. RP Shear actually invented the first rotary dye system for soft gels, and that same model or system still sits in the Smithsonian Institute today in DC. So I always think that's like a super cool little fact that, you know, the history of Catalent sits in the Smithsonian Institute. So since 1933, we have evolved and acquired different businesses throughout the years, really as a response to our partners' needs. So where we sit today, Catalent is a publicly traded company. We're part of the S&P 500. We have about, I think, close to 50 sites at this point worldwide and around 20,000 employees. So if you look at the buckets of technologies that we offer now versus back, you know, way back in the day, just with soft gel technologies, we have our oral technologies, biologic, cell and gene therapy, respiratory and ophthalmic, and consumer health. And then bolted on to all of those, we have our clinical supply services team. So the clinical supply services team is able to offer primary, secondary packaging, labeling, distribution, storage, and all of your clinical needs. So it can be a one-stop shop. The one thing that Catalent doesn't offer is API synthesis. So, you know, if that's something that someone would need in their program, we don't actually make API. So our models usually API comes to us, and then we can pretty much take it from there. One question Brett and I often get asked is, you know, when is a good time to engage with a CML? And of course, you know, you're going to have different phases of your program. So some people often feel like, you know, oh, we think it's too early. You know, we're not ready to actually talk with somebody yet because, you know, we don't know what we need. But Brett and I really live in that preclinical space. So we're oftentimes seeing people just, you know, 2 people in a molecule coming out of discovery. They're not really sure what they need next, but that's kind of what we're here for, is we really have a consultative role to try to walk through like maybe what that scope would look like, what that next step looks like, and beyond. So within the preclinical space, we do have some, we have DMPK modeling that's available, which is what Brett was talking about earlier, that first consultant that lives over in Sweden, and he's been with us since like I think GSK,
AstraZeneca.
AstraZeneca.
Yes.
So he's been doing modeling for like 20 years. So that's something that, you know, we see a lot of our partners on the early side that they really find that it's valuable. It can give you a good idea down the road of maybe which, you know, path your molecule may take. And then we have our solid state CP as well. But, you know, we can support anywhere through this continuum. So people sometimes don't come to us in the very beginning. We see tech transfers come in. We see people coming in for secondary supply later on down the road. So it just depends. And maybe they only need packaging. So they might not, you know, engage with us for every single offering that we have. It might just be, you know, piecemeal for 1 or 2 things. One thing I like to highlight is our Patient First initiative at Catalent. So it really inspires everything that we do on a daily basis. You know, whether it's myself in business development or the people who are actually manufacturing in the suite or, you know, science and technology team, it's at the forefront of everything that we kind of believe in at Catalent. So, you know, what does it mean to us? It means that every outcome matters. So it doesn't matter to us if it's a, you know, big pharma bringing in their project, or, you know, a rare orphan disease that maybe is going to have very small volumes and it's only, you know, supporting a few people in the world. That patient is still on the other end, and that's what really matters to us. So every outcome is very important. So we want to really make sure that when programs are coming into Catalent, that they're being set up for success, because there's someone on the, you know, the end user is someone out there who's going to need this medication that's going to be changing their lives or, you know, supporting their lives on a daily basis. And so to that extent, it's really important that we're helping to de-risk programs early on, because that way you've got a scalable, reliable process that you can use down the road. We realize that early on, it's really important to be flexible. So we are hoping that, you know, when programs come in, we can offer a tailored approach, because every program is different. So every molecule is going to have its own set of challenges. And we really recognize that. We've been doing this over 85 years. So we realized that, you know, we've seen a few compounds in our day, right? So we realized that there's going to be unique challenges. But I think one of the nice things about Catalent is because it has such an expansive network, they really have the breadth and depth of the scientific support that's needed. So there might be, a site that you're working with in St. Petersburg, Florida, and then maybe that team hasn't seen a particular challenge, but there might be someone in Weinheim who has seen that challenge before. So we're able to, draw on that pool of talent that we have to support programs successfully. And then, of course, we realize there's, speed. Speed is important in every program. Trying to get, either to your next funding milestone as quickly as possible. You're trying to get to your next filing as quickly as possible. So we recognize that as a company and want to make sure that we're, meeting your timelines as quickly as possible, but also, holding on to that really good science along the way. Last one for me just gives a overview of where Catalent sits in the world today. So two that I'll highlight just because they're new to the network is San Diego. So San Diego just opened our clinical supply services site that's on the same campus as the manufacturing site. And that's very similar to the way our Kansas City site is set up. So for small molecule, I support 7 sites within the US. So it's St. Pete, Somerset, which is our global headquarters, Malvern, Pennsylvania for micronization, Kansas City and San Diego, and then over in the UK, Nottingham and Haverhill. But if you'll look, typically the strategy within Catalent is that they're sister sites. So we usually have, not just one site that does one technology. There's typically another site within the network that also does that technology, and it gives you a safety net within the network. So if, God forbid something happens to one of those sites, there would still be a backup site within the network. And then we also have a new clinical supply site that opened in Shiga, Japan as well. So I guess the takeaway here is, wherever your program would sit in the world, chances are there's probably a site, Catalent site nearby that would be able to help support. And they each have their own niches. Not every site is going to do everything. Some are experts in particular technologies, and that's kind of how they were acquired. So they were acquired for that purpose. So we don't integrate like every single technology into every single thing. Yeah.
Thank you very much. You have a group who is working on small molecules targeting RNAs by NMR.
RNA.
So this is a question from me to you.
Small molecule RNA.
No, small molecules targeting RNAs.
I think what he's asking is if you have a small molecule targeting something like an RNA, like a nucleotide,
yeah,
do you guys have an ability to image it via like NMR?
Oh. Do you know that?
Yeah, well, we have a structural elucidation team in RTP, but that's as close as we come to it. They're more focused on probably compound identification, related substances identification. So yeah, that would be they'll do NMR, but again, it's usually columns.
NMR everybody is doing, but is doing for small molecules targeting proteins.
Yeah.
Here the word is very good.
Yeah.
We are doing completely contrary. Small molecules targeting RNAs.
Yep.
Everybody is looking like the crazy people, but if this would be developed in 2018, probably COVID story would be different because the world would have a lot of facilities to develop small molecules targeting RNA from viruses. So
is that something we could take back internally too, maybe, Anna?
Yeah, I think it's a little outside of what we do, but it's interesting to know though. It's Thank you.
Thank you.
Yeah.
We're to be clear, she touched on it. We're a drug product company. So we engage really, that said though, a lot of what we do is dictated by the properties of the drug substance. So we're a little downstream from the API itself, but we certainly are dealing with, so we can give feedback and there's things we can do to manipulate the API in some states, but that's really more a drug product company for us, what we do. So the goal of this presentation is really is not to drive down into the details, although some of the aspects I will go a little narrow and deep on, but it's really more to impart, I think, practical advice for research stage companies who are trying to transition off of the benchtop and move into human, ultimately human clinical studies And, what, as she mentioned, we know a thing or two because we've seen a thing or two. And what advice can I give you in terms of, decisions you make today, what they can do and impact you, not just immediately, but down the road. So I'll start by talking a little bit about the differences between what we're calling, or when we say things like early and late stage drug product development, what are the differences and how you should be thinking different from there and what's important. Also touch on some basic principles of oral drug delivery, as I like to say, why formulations matter. And then beyond that, looking at multiple, a lot of times when we're talking to research stage companies, they're looking at not just a single asset, but possibly a series of assets or multiple compounds. And so how do we look at those and just assess what I call the developability, not the drugability, which means one thing in medchem, but the developability or formulatability of a small molecule for oral dosing? How hard or how simple or how complex is this going to be? And then selecting, once you've kind of looked at that, how do you select the right clinical dosing strategy for your molecule, certainly from a formulation standpoint? And then I'll touch on just briefly on the end, how those early decisions impact your timing and ultimately your long-term costs as you transition from that early to late stage. Hopefully we'll have a little time for Q&A at the end. So early versus late stage product development. So I used this one last year and I love this visual because I think it's, a picture worth 1,000 words, and it says, and this is true, this is common to this is not just drug development, this is any new technology, any new discovery, any making something new in the world. Early on, there's the focus is to select the right path forward, pick the right track. In the late stage, you reach a point where you're on the right track or be on the right track, and it becomes all about making that path smoother. And how do we pick the right track at the beginning that gets us there, but also hopefully helps us smooth the hills out a little bit. And really it starts with recognizing that the differences between early and late stage really put you in a situation where you have fundamentally different organizational endeavors. In early stage, you're a truth-seeking organization. It's all about asking hard questions of your technology and being prepared to react and accept the results of those questions. You're all about efficacy, establishing the efficacy or lack thereof, reducing the risk, being loyal to the scientific method and experimentation. And I probably don't have to tell anybody in this room about this, but you operate at low fixed costs. Everybody wears a bunch of different hats. And at the end of the day, you're testing until you hit that point. There's an inflection point where you now have essentially proof of concept. And you don't just believe in your technology, but you have data to back up that belief. And that's when you make that shift from I'm challenging myself day to day to now I'm looking to how to make this a successful product. And there's a commercial side of this, and there's also the ethics side of it. The moral side. We're not making something to a handheld device. We're making things that will save people's lives and make their lives better. So we want to, how do we get this product to patients the fastest? How do we make it patient friendly? But there's a business side. How do we maximize the value? We believe in our product. We know our product. We're focusing on commercialization, and we're refining instead of testing. And so where is that inflection point in our industry? What is that? When I talk about that, what do I mean between early and late stage? And I go back to really a high level, a very typical general overview of the US IND process, which many of you may or may not be familiar with. And to touch a couple of key milestone points, from discovery at the far left to the point of going into GLP tox, where we're actually demonstrating in animals, usually a rodent and non-rodent species, safety at levels that are significantly higher than we're going to dose in humans. So we're looking for adverse events and understanding biological activity and safety. Once the IND is filed, we move into a huge milestone for many companies, which is the first time you dose this in a human, and that's typically small groups doing single ascending and multi-ascending dose studies, typically in healthy male volunteers, unless you're oncology, which you may be in patients, but often in volunteers. And here the focus is safety, to demonstrate safety. Once that's established, you can move into clinical phase 2, where you from a traditional standpoint, you first get yourself into patients. And again, the dosing groups are relatively small because you're continuing to evaluate you're looking for any side effects, any adverse events, but you're beginning to establish efficacy and evaluate efficacy of your product. And once you've gone through a series of phase 2 programs, and they differ from drug to drug, you really, to me, that's where the inflection point happens. That's clinical proof of concept. So when a lot of times in our team, we say clinical proof of concept, we typically mean the end of phase 2, where you make that transition into phase 3. And in phase 3, you're moving towards much larger patient groups. You may have diverse populations. You may be in multiple continents, multiple countries. You have to have statistically powerful patient groups, and you're moving towards developing the data that will justify the approval of the NDA. And there's a point where now you're starting to think about the commercialization. You're starting to think about more big picture items. And you know, a lot of times the small companies we're working with have no intention of ever going through this process. They look to partner their assets before this. But what I'm here to tell you is that some of these decisions you make early on can smooth out or cause problems as you make that transition. So, you know, back it up a little bit, looking at, you know, the stages of pregnant, and again, most folks who consult research, they're very familiar with this preclinical development from discovery to human dosage form. And you know, the numbers on the bottom, my big boss actually put this together a couple of years ago, and I've never argued with the numbers. I think they passed the eye test. If you go back to the earliest stages of establishing disease biology and finding, you know, molecule targets, and you go into high throughput screening and confirming those hits with second assays, you may be looking at hundreds of thousands of molecular structures, and you begin to narrow that down to the point where you're starting to what I call late stage discovery. And I'll go here, late stage discovery, where you begin to want to establish efficacy in a live model. You look at safety in a live model. You begin to do some of those developability assessments because you're trying to turn a dozen compounds into 1 or 2 leads. That moves into testing PK and dose range finding studies. And then you move here to the IND enabling studies, which are those GLP safety tox studies. And that, of course, leads into real developing a human dosage form and ultimately clinical studies. But this point here is where often the formulator, your formulation team or your formulator or someone like us gets our first telephone call, because that's where the formulations start to matter. Now we've got to take this drug, take this molecule, we've got to put it in a live subject. And how are we going to do that when the molecule is terribly insoluble or terribly unstable? And that's where your formulations start to matter. Yeah.
Should the formulation go before IND enabling development human target product? You are testing human target product, right?
So the short answer is you absolutely have to. It depends on how challenging it is from a. Let me go back here. So the formulations that you use to do these tox studies are not necessarily the same formulation that goes into the first in human.
They could be, but they're not necessarily.
Could be, but not necessarily.
FDA will question.
No. Not for oral. Now, injectable, it's a different animal. If you're going to take an injectable product in, for all practical purposes, the method of solubilization, the stabilization has to be representative from GLP to GMP. Oral, we see drugs are put in a maybe dissolved in a lipid-based carrier or something kind of fit for purpose, or maybe they use a cyclodextrin, get the data, and then they want something that's much more patient-friendly, much easier to dose. Maybe taste masking becomes important, or blinding becomes important. Maybe your clinical sites are not, they're not in the business of mixing up things on a day-to-day basis. They want the capsules, they want the tablets that you can dose. So those are usually the reasons that drive companies into making that change from what I call IND enabling. There's preclinical, there's early preclinical, dose range finding studies, efficacy, and then there's IND enabling preclinical, which is GLP tox.
Similar question. So if you don't have a good formulation, the time is doing the efficacy study in preclinical, whether they will really compromise your data in terms of efficacy, or we should do the formulation, even the preliminary formulation to get in the right.
Depends on what you're seeing. So here's the problem. And that's good. It's a good segue to the next slide, because usually this is when we say formulations matter, because a lot of times this is not ripped from the headlines. This isn't real data. I think we made it up, but it's representative of the type of problems that are brought to us by customers. They're saying, well, I'm dosing my, let's say it's a 50 milligram suspension, or maybe it's a 50 milligram in solution in like a Labrisol. And I'm seeing much lower than expected. I thought my curve was going to be here. This is plasma concentration versus time. But look what I'm getting. Look at the big error bars that we're seeing. Why? Why am I seeing that? I'm just doing that for efficacy in mouse. Why am I seeing that? Or the half-life is too short. What am I doing? What's going on? How do I fix that problem? So you may have to fix that very early on. Now, the way you fix it, the way you address it may be fit for purpose. It may be a kind of a simple get me through mouse, get me through rat type formulation. It doesn't have to be the thing that's going to carry you. And I'll talk about technology platforms in a few minutes. It doesn't have to be the most advanced technology platform that's ultimately going to carry you into human studies, but you are going to have to address it. What if you see, what if I see differences between species? Look, here's my rat, that looks pretty good, but what's going on with dog? What is this? Why is this? Why is this happening? And is there something we can do with a formulation to maximize bioavailability, but smooth out variability as well? How do we do that? And so that's usually where we're kind of getting our first calls. That's usually the problems that we're having. Does that answer your question? I didn't know if does that answer your question? Okay, making sure. So to talk about that in a little more detail, let's talk about some of the principles of oral drug delivery. And I think most people understand this, I think, but just to point out that what is the first thing a drug sees when it's ingested? And of course, it's the stomach. The first thing it goes to the stomach, where the pH is relatively low. And a lot of times what we'll see is for the weak base compounds that we see a lot in pharmaceutics, you actually might get something along the lines you see with this blue curve, which is the drug is actually quite soluble in low pH. And so there's every reason to believe that drug is going completely in solution in the stomach. The problem is that the stomach has very low surface area and very few drugs have any appreciable absorption in the stomach. The drug has to pass through the pylorus and into the proximal small intestines, where the pH goes rapidly up. And there in the duodenum and the jejunum is where most absorptions are going to occur. So when I ask you about how soluble your molecule is, I want to know how soluble it is in the intestinal media, not in pH, not in stomach, and not even necessarily in the vehicle that you're trying to dose it with, the solution you're trying to dose. Is it going to stay in solution and not do what this blue line, it went up, it was great, but as soon as it hit this higher pH, it precipitated out. Now, some of it is being absorbed. Some of it is being absorbed by the gut wall, but is it being absorbed fast enough to offset the precipitation rate? And so what this actually came from one of our spray drying presentations, and what we're trying to do with concentration sustaining formulations, or I guess you could probably argue if the precipitation rate wasn't fast enough, this is called the spring, and this we think of as a parachute. Do we have a formulation, or can we come up with a formulation that is going to keep it in a supersaturated state long enough to let the disappearance of this drug, this is local concentration, the disappearance of this drug is being caused by absorption through the gut wall and not precipitation. And I'll talk a little bit about how we measure that. But that's what we're thinking about when we're looking at what's going on. And of course, the last thing it sees is the colon. And the point I would say about that, the colon is, when you start thinking about short half-lives, and you're often asked about sustained control release or sustained release, it's important to realize that there's very little things that we can do to slow down motility. And so the drug is going to have to be absorbed in the colon. And so a couple things are happening in the colon. Much lower surface area. So compounds that are not very poorly permeable to begin with are probably not going to be absorbed in the colon. The other problem is solubility. The pH is still going up, and the volume of solution to solubilize it is going down. So it becomes much more challenging in the colon. So ideally, you've got kind of about a 4-hour window for immediate release drugs that you really want to focus on hitting. You want to maximize the solubility in that space. Is there a question? I thought I saw. Do you have a question? Oh, okay. Sorry. So one thing that's important to think about is understanding the pharmacokinetics, and really what we think about as formulators. What can we do to focus on the absorption of the molecule, to improve absorption? We can't. What do we do about distribution? And ultimately, the drug. What the drug does, what the body does to the drug, it metabolizes, eliminates it. But we want to start thinking about this in formulation because there's a relationship between the concentration and time and the drug effect. And what can we do as formulators to focus on or impact this? And so some of the questions we're asking early on is, what is actually driving your efficacy? Is it a Cmax effect? Is that all it is? Or is it an area under the curve? Or is it time above a minimum effective concentration? So you kind of need to go back and really get an understanding of your ADME property, so you can understand its half-life, and come ideally you're going to have some feeling and understanding about what the pharmacodynamics of your product is. So it's not strictly, as formulators, we're thinking about we focus on the pharmacokinetics, but understanding that pharmacodynamic target is important for us and establish what our goal is. So thinking about that, how do we assess the developability of small molecules? So we look at small molecules all the time, and we say, well, how do we pick the winner among a source? And I don't know, just show of hands, I'm sure a lot of people have. Has anybody heard of the BCS? It's been around for years. BCS, bioavailability classification, BCS. Okay. All right. So I say that because it's usually a starting point. If you've been in formulations in the past, I'd say 20 years, you've certainly heard of it. And it was actually developed, the BCS, biopharmaceutical classification system, it's just it's a way to categorize molecules from a bioavailability standpoint. And it was proposed about 25 years ago. I think it was Dr. Amadon out of the University of Michigan. And it's important to remember, we as formulators talk about it all the time, but it's important to remember that it was a regulatory tool. And the concept was that if you have a generic drug for which it is highly soluble and good permeability, formulation really doesn't matter. So I could go and copy that generic, and I really don't have to be terribly concerned about the details of that formulation because it's going to be orally available regardless of the formulation. So I may be able to apply for it and receive a bioequivalency waiver or a waiver on bioequivalency study. So it's really a regulatory tool. But formulators immediately latched onto it. It was a great way to think about this. Wait a minute. I've got 4 quadrants. I've got good soluble, good permeability. I wish all drugs were that. They'd be easy to formulate. About 70% of molecules that come out of discovery fall into category 2, which is hydrophobic, small molecules with decent good to decent permeability when in solution in the gut to the gut wall, but they are poorly soluble, brick dust. And then, of course, Cat 3 would be good solubility but poorly permeable. So you'd have a hydrophilic molecule. Here, great solubility is not a problem, but are you going to be absorbed by the gut wall? Start thinking about IV with drugs like that. And then Cat 4 is the worst of all worlds. Not only are you fully soluble, but even if you can have it in solution in the stomach, or excuse me, in the intestines, you have poor or very slow permeability. And these can be very, very challenging to formulate if they fall in here. And then there's the gray areas. Where are you at right along the line? So that was all wonderful to think about. But about 10 years ago, there was a paper by Butler that proposed, that BCS is fine, but it doesn't really help us tell us what we need to be thinking about or how to formulate. It tells us what we're up against, but it doesn't tell us how to formulate. So they proposed, they built off of the BCS, and they made a couple of key changes. And I think the most important one was, one of the most important was, first they said, not only we want to understand solubility, but we want to understand solubility where it matters. So what we want to really look at is what is the solubility in intestinal fluid. So that's fasted state, simulated intestinal fluid, and fed and fasted. But so when we start to ask these questions about how to rank a molecule in the DCS, ideally we know the solubility in intestinal media, fed and fasted state intestinal fluid. If not that, at least intestinal pH. That's what we want to understand. The other thing is, the y-axis is permeability, but we want to quantitate it. We want to give it a good number. And what the BCS uses is human PF, estimated human PF, which is usually derived from Keiko studies, although we have some software that can make pretty good in silico predictions of human PF. So that's your y-axis. And your x-axis is not just solubility, it's the dose to solubility ratio. So it's dose divided by solubility. It's recognizing that solubility, the challenge of solubility, is relative to the dose you're trying to achieve. And I'll highlight that in a minute with the next slide, but I'll point out that the paper went on to propose a way to calculate what's called a solubility limited absorbable dose, or SLAD, the SLAD line. So at any given PF, you can calculate what in theory would be the maximum milligrams of dose that would be soluble in the human intestines. And The animal, the living GI tract is much more dynamic than this, and none of these numbers are ever written in stone. I call this a ballparking exercise. What ballpark am I playing in? And the concept you can see at higher PF, the reason this line is slanted is because above a PF of 1, that's the cutoff line there, you have, it's a rate of permeability. It goes back to that parachute effect. Are we absorbing drug fast enough to keep up with the potential for precipitation? And so here's the example of why that matters. So we have, I talk to people all the time and say I have a very poorly soluble compound. I have a poorly soluble compound. What about that spray drying stuff you guys do? You know, let's go spray drying. Wait a minute, hold on. Maybe you need it, maybe you don't. Because we also talk to customers who have drugs that are highly potent, and the therapeutic dose may be milligram quantities. In some cases, every now and then, sub-milligram quantities. So while you may have what on paper is a poorly soluble drug, if it's 5 million, in this case, and whatever the solubility value was at this given PF, if I go dose divided by solubility, it's 5 milligrams I'm here, at 50 milligrams I'm here, and at 500 milligrams I'm here. So the flip side of that is we also have customers that have drugs that are not terribly insoluble, but they're trying to dose 7, 8, 900 milligrams, a gram and a half. And so now they have a solubility problem. And what this tells me as a formulator is a couple things. If I'm over here, I can probably take the native API as it is, and I could literally dose it in a solution, probably right on the spot. I could put it in water, solubilize it, and give it to my patient, let them drink it. That's a very easy clinical first in human strategy. I could just put the API itself in a capsule and just give it to the patient. I could blend the API for reasons I'll go into later. I can do a lot of different things. I don't have to be so worried about the particle size distribution of the API. Surface area is not important. It's well in a soluble location. When I'm in 2A, I'm inside. This is called 2A and 2B. I'm inside the solubility limited absorbable dose line, but I'm in the 2 category. And the problem there is it's not a solubility limit, but it may be a dissolution rate limit. So there, when a dose and a solubility falls into that range, we start thinking about things to increase the rate of dissolution. Maybe we add a surfactant. Maybe we pre-dissolve it. That's where pre-dissolved formulations are very helpful. Maybe we, long-term, you go back and find a better salt that improves the solubility. And so there, and then of course, the worst situation is you're at a dose where you're in what's 2B, and if you're far enough away from that SLAD line, even if you have an acid soluble drug that goes in solution in the stomach, it's very likely that some of that dose is going to precipitate out. Now, where it falls, prior to absorption, but it was going to precipitate out and pass through the GI tract undissolved. So there's some gap here. Where exactly it is, who knows? Who knows? It'll depend. It may vary from subject to subject, but there's some amount of your drug that you're going to have to, if you're going to maximize this dose, if you're going to have all of this available for absorption, you're going to have to do something with this API to keep it in a supersaturated state in the GI tract. And that's where that's the first thought process there. That's the first thing we do. Very simple way of looking at it, the ballparking exercise. And also another good point is when I ask you about simulated intestinal fluid, I want to know fasted state, but I also want to know fed state. Because often you'll see usually positive, sometimes negative food effects, and you may see a shift. So in fed state intestinal media, this molecule is actually pretty close to the SLAD line and may not need any significant solubility enhancement at all. It may just work. The problem with that is you don't want to walk into a clinical study having to say, my patient needs to eat a cheeseburger. Because diets are different, patients are different, fed states are different. But what this tells me is that there's an advantage to being in a fed state or something like a fed state. Now, it could be a couple of different things. It may just be the pH is lower. It may be based on the pK of the molecule, you're fine with the pH. It may also be the bile salts. So when we start seeing this data, though, we start thinking, there are formulation strategies we can use to leverage that, and there are, that can smooth that food effect off and maybe take advantage of it and improve it. So what if you have most of the time we're running into our solubility enhancement. Yeah, go ahead.
So are you testing frequently the blood-brain barrier solubility? So
it depends if you're central nervous, what's your indication, right? If it's obviously central nervous system, yeah. So it's I don't have a test for blood-brain barrier solubility, but what we are looking at are some other characteristics that indicate that it's probably going to cross the blood-brain barrier. One of them is total polar surface area. So I think the cutoff on TPSA is around 90 angstrom. If you have a TPSA below 90, it's considered, I think it's Weber's rule, if I remember correctly. So you have the rule of 5, which I probably should, that's a good question because I should probably add that to this slide because that's a fundamental rule of 5 question, right? So how many hydrogen bond donors, how many hydrogen bond acceptors, how many 500, are you below 500 molecular weight? And do you have a log P, I think, of below 5? Those are the old school rule of 5s. There are hundreds of drugs, maybe thousands of drugs that do not meet those rule of 5s. They violate one or more of those rule of 5s. That does not rule you out as being an oral drug. You also, nor does meeting those automatically make you, because you may have metabolism issues. But there's also some other rules like the Weber's rule, which I think is TPSA, total polar surface area. There's something about, I think it's less than 10 I can't remember the number of rotatable bonds. So rotatable bonds and total polar surface area of less than 90 angstrom is considered conducive to blood-brain barrier crossing. 140 is considered the cutoff, or roughly the cutoff to oral bioavailability. So again, I see molecules that are orally available that are higher than that. But yeah, it's not uncommon to see. The other thing we're worried about as formulators is I can't do anything to make it more likely to cross the blood-brain barrier, but I can drive more drug into solution, into plasma. So the higher I get into plasma, I have a better chance of driving across there. Then of course you got PGP. So you got transport, you got efflux transporters, which I can address at the gut wall, but I can't address at the blood-brain barrier from a formulation standpoint. So there are, I think, really 3 platform technologies in industry today that check a couple of key boxes if you have a poorly soluble and or poorly permeable compound for oral delivery. And these boxes are important because they're technologies that are proven and scalable. They are available globally under GMP. So you have GMP excipients that work with them. And then they produce at the end, and most important, really, a patient-friendly finished dosage form, a tablet, a capsule, something patients can take. And they fall into 3 major buckets: amorphous solid dispersions, lipid-based drug delivery systems, and particle size reduction. In an amorphous solid dispersions, what we're doing is generating a solid solution of the API, what probably wants to be a crystalline API, render it amorphous through a couple of different techniques. Spray drying and hot melt are the most common, although we have some other techniques we can use. And we render it amorphous and stabilize that amorphous form with a polymer, with a stabilizing polymer. The polymers also typically provide concentration sustaining power on the back end, so you can get that parachute. You can do it through co-dissolving the API with the polymer in a volatile organic solvent like a methanol or an acetone, and flash drying in a chamber. And what comes out appears to be a homogeneous white powder, but in fact this is a solid solution of amorphous dispersion of API and polymer. The hot melt extrusion is another way to approach that, but you co-melt it and you extrude the molten API polymer molten system that immediately cools and it's chopped up into little pieces. And basically, again, it looks like a powder, but it's in fact it's not a crystalline powder. It's an amorphous solid solution of API and polymer. So 2 different ways to get essentially to the same thing. Lipid-based drug delivery systems are essentially solubilizing the drug, dissolving the drug, pre-dissolving it in a lipid-based carrier. And it can be true oils, it can be lipid-like surfactants, it can be some co-solvents in some cases, but essentially a lipid system. And what you're dealing with is a non-aqueous lipid carrier that can be dosed as a solution, but more often for ease of dosing and then long term, you're putting that in a capsule. They can be a 2-piece hard shell capsule, liquid in capsule, or ultimately a soft gel. For commercialization, you're heading towards a soft gel, use lipid based. The advantage here is that you have some, but it can do what this does in terms of these are usually self-emulsifying. So if you have an oil and a surfactant system, you can develop self-emulsifying or self-microemulsifying delivery systems, and you get the micelle formation, and essentially you keep the drug in solution as it moves into the intestines. So you get the parachute effect. You also are able, with certain types of excipients, in this case, in some cases, you can target PGP, so you can maybe set on some of the transporters. You can also you can also potentially as these are digestible formulations, and as they're digested, sodium caprate is generated, and you can temporarily open tight junctions. So molecules have less than ideal permeability. You may be able to improve the permeability of these systems. And then of course the third thing with these is that food effect. We'll make the food effect. digestible lipids, digestible oils, essentially you're bringing the meal with the dose. So you may be able to smooth out that food effect. And then the last one is particle size reduction, which is just micronization. You're increasing the surface area of the API, putting more of it available for dissolution. I would say that's more of a 2A, DCS 2A. If you fall in that category, that triangle, that's probably going to work. If you're in the neighborhood of that, that may help you. If you're far out on a DCS 2B, you can probably chop these up as much as you want, even to the point of nanomilling. At least you certainly have to be to a nanomill state where you can improve the solubility of a DCS2 or the absorption of a 2B compound.
Is that lipid-based reverse is a liquid or solid?
Usually liquid, but it can be a semi-solid depending on the excipients that are used. Yep.
What's sort of the order of preference of these different, all of them are viable, but what's, if you had like.
If this works, that's the one you want. That's the easiest.
Yeah.
Just micronize your API.
Mhm.
Maybe you can even get your API produced. Maybe it can be crystallized in essentially a micronized state. But that's the easiest is the preferred.
Mhm.
But if you have a permeability problem, like say let's not say a DCS4, but say you're a DCS 2B/4, like you're in that gray area, and maybe depending on the test subject, then maybe this is your preferred route because this may help you improve the permeability and even permeability of the molecule. So it improves your bioavailability. So your solubility.
So if all this technologies are proven, how do you resolve IP? And if you have a formulation for your drug, how do you protect it?
So, well, good question. We get asked about, I'm going to go off subject here a little bit. So we get asked about, can you give me a formulation that I can patent formulation? And there's a couple of things to consider in that. I think the biggest one to me is that, there's kind of three major patents, and I'm not an attorney. I'm just going by, I'm a layman. There's three major aspects to patenting, right? So there's matter, patent of matter. So if you discover the molecule and you patent the molecule or salts thereof, you have a 20-year patent on that molecule. So ultimately, patenting the molecule is your most bulletproof, your most powerful patent. There's also the patent of use. So you deal with like 505(b)(2)s where folks come in with molecules that have been around. They have no IP around the molecule itself, but they're applying it to a new drug, or they're maybe coming up with, it's not even the formulation. It's just, we have to reformulate it because we're going to a different route of administration, or we're going to a different test subject group, maybe a different concentration. But you have an approval use patent, which may give you, I think, down to 3 years, but it can go 7, it can go higher if you're orphan, if you're pediatric. So a lot of times the ideal world would be to do an orphan pediatric, right? Because you get the longest. And then of course the third is actually to have a formulation patent. And I think the general, what the attorneys say to me is that's the weakest. That's the weakest patent. It's the easiest to get around. Because you can say, okay, I patented the formulation, but what if I can do the same thing on an old molecule? What if I can get the same therapeutic effect with a different formulation? And it's highly unlikely that you're going to come up with a formulation that's going to be, that it can't be, there's not some other answer in the world. So you have to start thinking about it. Is that a broad formulation patent? And and at the end of the day, the patent, again, this is a layman speaking, I'm not an attorney, but the way I was taught was a patent is a sword, not a shield. So there's nothing that says someone cannot infringe on your patent. They can absolutely infringe on it. The patent gives you the power to go prosecute them if they do. So it's on you to go after them. There's nothing that says that I can't, it's not a shield. It doesn't protect you from being infringed upon. So, if those are some of the questions you have, I urge you to, talk to patent attorneys, because they're the ones who can help guide you through it. But that said, we're formulators, and if you come to us and say, I want a patentable formulation, well, we do contract service work. And at the end of the day, this formulation that we come up is yours. And if you want to patent it, it's yours. Go patent it. But it'll be on you to go defend it. So I think that's our broad position, and there's exceptions to every rule, but that's kind of our position. And so usually it's actually the opposite. We're dealing with a lot of discovery companies who they've got patent matter and they want it as simple as possible. So it's actually the opposite. They're like, give me something simple. Don't make this anymore, because the cost of manufacturing, I'd say, goes up in that direction. and both small scale and long term. These are more specialized equipment, specialized in process engineers. They're scalable, they're proven, we can do it. We do it from spray dryers that are this tall to spray dryers that are 4 stories tall. We can do it, but make sure that's what you really need. So, bioavailability, more than just dissolution. This is an important concept because percent F, the overall bioavailability that you're seeing in those IV versus PO dosing, it's a function of the FA, which is the fraction absorbed, times the FG, which is the fraction surviving gut wall metabolism, and times FH, which is the fraction surviving hepatic metabolism. So the FG and FH taken together is the first pass loss. And so that's what the BCS doesn't take into account. It's a 2-dimensional consideration. You may be a 2, you may be a 1, but if you have a very high first pass loss, then you have a challenge. And there's not a lot that a formulator can do about it. So a lot of times as formulators, we're thinking about FA. What can I do to maximize this? I've got a poorly soluble or a poorly permeable compound or both. And what can I do? And we actually have a lot of tools that we can address solubility. We can address certainly solubility and maybe even permeability. We look at salt forms, these precipitation resistant formulations. Maybe you just have more frequent and higher dosing, more frequent and divided dosing in your preclinical studies. Maybe it's easier just to kick that formulation can down the road and just maybe dose more often. And of course, we have lipid formulations where we may be able to improve the permeability of the compound. But what can we do with FG and FH? And there we start thinking about there are some alternatives from a formulation, which you kind of have to fit the mold. We can target lymphatic uptake with our with lipid-based delivery systems, but there's a couple kind of these are no rules are ever set in stone in science. But roughly speaking, your molecule has to have a really high, has to be lipophilic. It has to have a high log P, good solubility in long chain triglycerides or long chain fatty acids, oils. So it has to be highly soluble in oils and a relatively small molecular weight. And we don't do them in house, but you can do measures of transport into chylomicrons. And if you check all those boxes, you actually may have a pretty good case for targeting lymphatic delivery. And we do this. We've got our St. Petersburg site, which is our lipid team. They will develop lymphatic targeting formulations. Little difficult to predict how they're going to perform, and ultimately you have to go into iterations of animal dosing to see. But in certain cases, you can see a significant improvement over first pass loss with that. Then, of course, the other option is if you can get pre-gastric absorption or an orally dissolving tablet, which would avoid the portal vein. Again, though, here's the problem: your residence time here is very short. You have a small amount of buccal mucosa, so the drug has to be soluble and rapidly absorbed prior to going down the esophagus. And the truth is, most of these orally dissolving tablets, even ones that target pre-gastric delivery, unless they've got some sort of bioadhesion, which we don't really do very much of, and that's that's something you could do if it came down to it. You can look at something, or you can do a lozenge. The challenge there is that the reality is with most ODTs, the bulk of the dose actually goes down the throat, goes down the esophagus. So, but that said, if you've got a very high first pass loss and you change that to very low first pass loss, your dose requirement actually go down a lot. So those are things we can look at. And how do we know that is through PBPK modeling. We were talking earlier about our PBPK team. Jan Nielsen is in Sweden. He's ex-AstraZeneca. We actually had another modeler, but he had to leave us for family reasons, and we're about to hire another one. But we have a PBPK modeling team that is that uses GastroPlus software. And this is important for us because what it does is, so I'll tell you how it works. So customers will come to us and maybe they've dosed mice, or maybe they've dosed rats. They've dosed a suspension, maybe they've dosed a solution, and hopefully they dosed IV. And so we'll take physical chemical properties of the molecule. GastroPlus software is a DMPK modeling. It's kind of the industry standard, it's the industry gold standard. Most big pharmas use it. There are other softwares, but this is the one we use. And essentially with the model, you can, in theory, input all the phys chem parameters of the molecule, in vitro ADME information, like if you have in vitro metabolism, hepatocytes, microsomes, in vitro permeability, Keiko studies. Maybe you've done some biodistribution measurements with your IV. Maybe you've got some blood blood plasma ratio data. If you can put all that into the software for that given molecule will give you a projected, this is the blue line, say the blue line, will give you a projected concentration curve at a given dose in the species. So what we'll do is we take the IV data, put it in, we put it in the model, and we really compare it to what we're seeing, compare the model to what's actually being observed. So, does that predicted curve actually fit the actual data observed? Sometimes it does, like this looks great. Sometimes it doesn't. And often it doesn't. And then what happens is the modelers in the model will do a parameter sensitive analysis and look at, what is really, what are the inputs wrong? Where do we modify the inputs to make the model fit the actual observed data? And then now you've got a model that's been refined for one species, and you can take that model and apply it to the next species. Let's say we'll go to rats. Is that refined and optimized model predicted for the rats? And sometimes it is, and sometimes it's not, because there are interspecies differences certainly. And if it doesn't fit the rat, we go through another iteration of parameter sensitive analysis and look at what is not working there and make it work. Then you go to dog, maybe you go to non-human primate. After these models have gone through 2 or 3 iterations of species, we usually have actually a relatively robust model. In some molecules are more challenging than others, but with most molecules, you have a relatively robust model that becomes pretty good at predicting what you're going to get in the next species, be it a non-human primate, a dog, or a human, which is usually what we're doing at the stage of the game. Now, we do this early on at preclinical. This is not why we do it. We don't just, it wasn't why we originally do it. We brought this capability on board because when we go back and we make those amorphous solid dispersions or those lipid formulations, and our customers will take those and put those into small animal models, because that's what they can afford. That's what they can do quickly. They'll put that into a mouse and we'll come back with data. Anybody can calculate %F. Oh yeah, it improved bioavailability, but is that going to improve it in the next species? We don't know. But if we have this model in place, we can now take the input, the PK inputs from enhanced formulations and predict how they're going to behave in other species. But what we learned really quickly was, and I would say we even applied even more now than that, is we use it early on to understand, are we really fighting the right fight? Are you really up against a solubility issue? Are you up against a permeability issue? Are you up against a metabolism issue? And the truth is you may be up against all 3 of them, but one may be weighted more than the other. And those are the ones we want to focus on as formulators. So that's where the PBPK modeling early on comes in. So I would urge you, to take that in what I call the in silico modeling approach very early on with data that you can collect. I have a couple of quick case studies. These again are more or less made up data, but they're very, they're true to what we often see. So here's an example. If I have a drug that is, let's say compound A, compound B, and I'm seeing a %F of 21%, 21% here. So almost a drug. There are marketed drugs with lower bioavailability than that. So we've got almost a drug, not bad, 21% when I'm just dosing it as a suspension. And we go through the PBPK modeling. The only way we can do this is go through the PBPK modeling exercise. We discover that, in fact, the fraction absorbed here is 25, and we've got very low first pass loss, or really reasonably low first pass loss. So you've got 85% surviving first pass metabolism. So that 21 is a function of 25% absorbed, and then 85% of that surviving first pass. The bad news is we're losing about 3/4 of our drug through the intestinal tract. It's not being absorbed; it's passing through undissolved. And then we have this other one where it turns out that it's the opposite. We have 85% being absorbed. Maybe it's decent solubility, maybe it's decent permeability, but we have a high first pass loss. We're losing about 75% in first pass loss. Which one of these is the one? Because you may look at this, if you had another PBPK modeling, you say, this is my more soluble drug. Why wouldn't I take this one? Right? And this is the one. But what we're finding, but in reality, addressing this problem is far more complex and unsure than addressing this problem as a formulator. I can work on this one. I may not be able to work on this one. So that's the so start thinking about backups with that one. Here's another example. Little busy slide, but I kind of like the point at the end. So we're looking at multiple molecules here in rodents, let's say rat or whatever rat. And if you're just going strictly by in vitro potency, you would say molecule 2 is the clear winner because it's a much more potent drug. And in fact, the percent F that we're observing in dosing, 33, 37, and 36, it's lower, but it's more or less in line with molecules that are the other potent molecules. So why not molecule 2? But then we take a closer look and we see that, well, we see that the amount surviving gut wall metabolism is actually quite low in molecule 5. Although it's a relatively soluble compound, you lose a lot of it in first pass loss. So that kind of eliminates it right off the bat. Molecule 1 is a class 4 with reasonable first pass loss, but it's a molecule 4, so it's always going to be a trouble. And then molecule 3 has incredibly high first pass loss, so it's gone right away. So which one's better, 1 or 2? And I would argue that because if you look at the first pass loss as being relatively similar, on the back end my question to you would be, if I fix this problem 2B, and I can fix it with an amorphous dispersion, a lipid-based carrier, something like that, what do I get? It's a little bit it's a higher bar to jump over than 2A. 2A, I might just be able to micronize it. Back to your question, which one's preferred? I can micronize this, or maybe we can do a salt screening and find a salt. So just the API itself is more soluble. And now we're going to have, okay, granted, if I fix that, the bioavailability is lower than this, but is that not close enough? I'm within 5%. And I could save myself, probably double, just on cost of goods. That's a rough thing to say. It's not necessarily right, but my point being is that that's a lot easier to fix than that. And if on the back end fixing that gets me here relative to that, I'd go that. I'd go there. So all that in mind, selecting the right clinical dosing strategy for your molecule. So what do we think about? And I want to pause here and say, let's not forget the patient. Patient is very important. The indication being the reason to treat the patient, and also thinking about mode of action. So the indication is, it informs the likely state of the health of the patient, other meds they may be taking. There's certainly the business aspect, the market opportunity, but also are we looking at oncology patients? Are we looking at pediatric patients? Are we looking at geriatric patients who may have swallowing issues? They may be on a bunch of different medications. I guess the second point is, are we going into volunteers or patients? And then when we think about mode of action, certainly, the small molecule, are we targeting an enzyme? Are we, specific targets and the biology? But it also helps us understand, more importantly, where the targets are, where exactly in the body does the drug need to be delivered. So like the question about the blood-brain barrier, the things we can do to push more drug in. And I highlight these because these as formulators are kind of the ones that matter. So I want to know early on, am I targeting this for a pediatric population, just as a regulatory tool to because I have to have a pediatric approval, or am I really going into pediatrics? And then that leads into this slide, which is, I won't spend a whole lot of time on this one, but it's remembering that pediatrics, children are not tiny adults. They're different. And that's not just titrating the dose, but it's also what's acceptable from a delivery standpoint. So we need to start thinking early on, and even if your first round is going to be healthy male volunteers, how quickly do you have to get in children? And so some of these, the application of different ones of these, solution versus drops, emulsion, suspensions, effervescence, powder, multi-particulates, I can incorporate in multi-particulates, I can incorporate an amorphous solid dispersion strategy, but I want to think about that early on because I need to know if I have to go that route. Tablets and capsules. So, you guys are going to get the slide deck. So I put it in here because I thought you perhaps may find it interesting. It's, it's from a European guidance document. But we refer to this a lot because a lot of times we're questioned about developing what's appropriate. And the question is how young are your patients going to be? Because if they're 2 to 5, if that's the youngest you're going to be as a 2-year-old, I have more options than if you're actually a neonate. So it can be complex. It can be quite a challenge. I'm going to start wrapping up here with oral dosing options for first in human studies. And at the end of the day, you've got to get yourself into humans. And most people are looking for kind of the most straightforward approach. And there may be there may be reasons. If you're oncology, you might want to think about For phase 1 is really phase 2, and phase 2 is really phase 3. So you may want to be a little bit more sophisticated dose form, but often you're looking to, what can I do to hit that first milestone? And what can I do to get there? And what are my options? And so I'm going to highlight all these in a little bit more detail, what they are and how they apply to the different DCS situations. But broadly speaking, they're what we call, to define my terms, drug in a bottle, DIB. Some people call it PIB, powder in a bottle, but I don't like to do that because the powder, what is the powder? Is it an amyl facade dispersion or is it a blend? It's DIC, drug in bottle, neat drug in bottle, neat drug. In that case, you may have I'll define it more. You may have neat drug in capsule, LIB, which is liquid in bottle, and that could be a solution, but it could also be a suspension, a liquid in bottle for ingestion, liquid filled capsules. I differentiate them from liquid filled soft gels. In principle, they're the same type of formulation, but the liquid filled capsules give you a little bit more flexibility for early stage. I'll talk about that in a minute. 2-piece hard shell. Formulated BIC, blending capsule, which differentiates it from the drugging capsule. And how complex that formulation is depends on what you need. And then formulated tablets, so they could be coated or uncoated. So drug and bottle. So what we're talking about here is neat API shipped directly to the clinic and then suspended or diluted upon administration. So love to work with you guys. Be fantastic. You may not need us for phase 1. If the drug is soluble and you don't have a taste issue, it suspends relatively easy. There are plenty of studies that go straight with essentially a container of API to the clinical pharmacy, and the pharmacy reconstitutes it or suspends it at the time of administration. It will come down to your clinical strategy, your clinical protocol, what you can and can't do, and also the chemistry of your drug. It gives you a lot of dose flexibility and allows you that extemporaneous preparation. Issues, if you have an issue with taste, it could be a problem. They're healthy male volunteers maybe, and they're usually grad students and they can drink anything, but the gag reflux is real, and if they spit it all over the floor, that's a problem with your clinical protocol. You may want to think about micronization. It may not completely preclude you from some sort of formulation development, and if it's you may still have to go micronize and pick the right particle size for suspension. And then you need to think about, perhaps you even want to get a little bit more, I think you're starting to lay the formulation or cross over the formulation. If you start thinking about, what if I do a cyclodextrin formulation solution? What if I have something in the solution that solubilizes a little more? Now you're really in a formulation. But, maybe it's a hybrid there. And the one thing you have to be careful with, maybe you have a drug, or you have a toxic drug, or a drug that cannot be openly handled, you may have to take a precision weighing strategy where you actually have vials with the exact amount of API in it, where they're stopped with the clinician can reconstitute it using a syringe, fill it in, get a solution, make it safe, and then it can be injected by the patient. In those cases, clinical pharmacy is not going to help you. You're going to have to go somewhere and have a decision field. The drug capsule strategy is really not too take in. So you can maybe push it down a little bit, but you're not really compacting. You don't want to compact, you'll break the capsules. And so maybe what was a 0.2 or a 0.15 grams per cc, maybe you get the effective fill down up to like maybe 0.25. Realistically, you're not going to put more than about 250 milligrams of neat API in a double zero capsule. And for those of you who aren't familiar with it, think about a multivitamin or a fish oil capsule. That, they're kind of big. So, a geriatric oncology patient may not be willing to do that. Now, the other problem is what you can do, a manufacturer can do, is be precise, if not accurate, with this strategy. You may say, well, how do I know I'm getting the same amount in? You'd be surprised. It's actually quite precise. The precision's good. But what I cannot do is I can't be accurate. If you tell me you want a 200 milligram dose, what you may get based on your density is a 185 or a 205 or a 210. Now you'll get that 210 over and over again, but that's what you're going to get. And then you step down to the next size and you're going to get what you get out of that. But the thing is, that's usually okay in phase 1. It's usually close enough. So, and if it's not, then you have to go to what is the next thing, which is, well, it's not the next thing. I'll talk about it in a minute, but you have to go to a formulation. If you tell me I really, really have to have 75 milligrams, if maybe I can do it this way. And if for some reason I can't do it that way, I can blend it. I can blend it to a ratio that's going to give me that same volume every time. So just understand the limitations of it. Someone says, "Oh, go drug and capsule." Probably may work for you. Do you have a DCS1 or worse, a DCS2, 2A? Excuse me, a 2A. And then what are your dosage strengths? What are you trying to hit? So it's possible, but it may or may it depends. Liquid and bottle. I'm not going to spend a whole lot of time here because essentially you take the powder and bottle, but pre-do it. And there may be reasons you do that. Maybe you really want to have a little more sophisticated formulation. Maybe you want to have a taste masking, but it requires development analytical and careful analytical support because stability is always going to be even more of a challenge there. But you can produce bulk liquid in bottles or in unit dose bottles. But I'll focus on these two a little more. Liquid filled hard shell and soft gel capsules. So in this case, same thing. The API is dissolved, or in some cases suspended in lipid vehicles, which can sometimes work. If you don't have sufficient solubility in a lipid vehicle, but you get that positive food effect, it may be valuable to do a suspension of the API in a lipid vehicle, fill that in a capsule, because you're not really solubilizing it so much as giving it that food effect advantage. BA improvement through the enhanced solubility and in some cases permeability. Anytime you go to these unit doses, be it the drugging capsule, blending capsule, tablets, liquid hard shell, you're set at the dosage strength of that particular batch. And if you need a lot of flexibility, you're often going to have to manufacture 2, if not 3 clinical batches, 3 dosage strengths, which increases your cost. The advantage of these over those is that it's a little easier to DR coat, delayed release coat these. Although we can delayed release coat those, these just are inherent. We can do that better. And the option, the difference here a lot of times is what we may be able to do, what you may want to do, talking about speaking of flexibility, is you may identify the lipid formulation vehicle and you can bulk prepare it and ship it to the clinical pharmacy. And then they can fill these capsules on site. They can perhaps even fill. So I said you're stuck with your formulation. You may actually be able to fill by weight. So you can modify your weight and target your dosage because you've got API in solution now, and how much you want to put in here, you can do, and that can be done on site. So that gives you that type of flexibility. Whereas here, you need to kind of prepare those ahead of time. Although there's always an exception to everything. We actually have the ability and have done to send air fills of these. You can send air fill soft gels to a clinical pharmacy, and we actually have a little video of how to puncture it with the vehicle, fill it, and then use a little, I think it's a Bunsen burner, or it's a heat probe they use to actually seal them back up. So, there are reasons you might want to go this route as opposed to this route, but often usually that kind of quick clinic, they're using this, and then if that formulation grows up to become this one day, you're not going to make 1 million of these. The manufacturing process is too slow. If you've ever seen a soft gel facility, it's amazing. You mentioned the RP Shura. The RP Shura invented the soft, they invented the rotary die process 70 years ago or whatever, and it eventually hasn't changed. It's amazing. It's like a machine gun. Just these things are pouring out, but it's a little complex, straightforward but complex, and mass producible. Then finally, the blending capsule. Here you're starting to think, okay, look, looks the same, looks white powder, but does that need API? In this case, it maybe is spray dry dispersion or a hot melt extruded dispersion, and it has been further blended with a variety of excipients that do different things. There may be super disintegrants because these STDs and HMEs perform a lot better when they disperse properly in aqueous media. There may be flow aids that help it to flow out well. There may be anti-adherents to keep it from sticking, particularly when using the automated encapsulation processes. These blends of powders have to flow through the hoppers and into fill dosing discs, and they have to do that consistently. So you're doing things, you have functional excipients in there, and you know that all has to be worked out. And it can be simple. It can be a simple binary blend, or it can be a more complex formulation. Again, you're looking at 2 to 3 dosage strengths. And these can be precision weighed, but usually often they're either flow filled or fully automated for GMP. And then of course you have tablets, which are essentially the pieces. A lot of times when we're doing these with STDs or HMEs, we're doing it now for a few thousand, knowing that we're going to transition to a tablet formulation. And that may be why you have some of those functional excipients. You may not actually need a flow aid right now to do that. You may not need an anti-adherent to do that, but when you get over to tablet punch presses, you're going to need all those. So get those in your formulation early, particularly if your clinical path is going to be relatively quick. So again, it goes back to those kind of making those decisions early. It might make you maybe spend a little bit more time right now, and it may save you some regulatory headaches down the road. And this is a table. I'm giving you guys. We show this to customers all the time. I'm just, you'll have these decks, but it's way too busy to try to read it all. But it essentially says what I just said. It has all the things that we have here, what they are, proximate range of what you'd expect to get in a single unit dose, the advantages, the disadvantages, and I just highlight the DCS fit. What works. Where are you on the DCS? Metabolism out the door. We're not thinking about metabolism right now, metabolic stability, but just pure DCS. Where do you fit? So please, you use that, and I'm happy to answer questions. And usually, what we're trying to do. So this is a table that I kind of want to. If I don't technically fill it out, I want to start to mentally fill it out early on in trying to make decisions. What is my indication? What is my site mechanism of action? How often do I need to dose it? How high does that dose need to go? What's the efficacy driver? Is it AUC, Cmax, TO, minimum effective concentration? Then start thinking about, we're somewhere up here right now. What are our patient population? Rat and dog. When does that have to be there? What's that dosing strategy going to be? How much of that are we going to need? And then any other special considerations. But then you get into, the patient population. Is it patients? Is it healthy volunteers? Phase 1, phase 2, phase 3. And again, that formulation can evolve over the course of this table. But if there's things you can do early on to make that transition smoother at a reasonable cost and a reasonable time frame, then that's what you want to think about doing. And then I think it's the last slide. Finished dosage form development manufacturing. This is just kind of a gut reality check. Because what it will say is once I've identified that platform technology, what realistically am I looking at? Because I'll tell you, back to some of the questions about the preclinical, it's not unheard of. You may have to go do spray dry dispersions just to get that tox data. It may be that the drug is too poorly soluble and there's nothing that's going to work. And you may be because there may be lipophilic vehicles that get it in solution at low levels, but how much oil can you really give the dog before it starts throwing up? So there may be needs to go do that work early on. So my question, the start point here is more of, I've identified my technology. I know it works. How long is it going to take me to get to the point where I have a bottle of tablets or capsules in a clinical pharmacy ready to give to humans? Release tested, stability in place, everything I have has got a label on it, and I can give it to the patient. How long is that going to take? These are conservative estimates. We sometimes can move faster, but I feel like that I won't be any worse than this. So I'd rather tell you the worst case and move quicker from there. But what you've got to remember is you can identify this, you can have these non-optimized dosage forms, but to get into a human, we have to go through a formulation exercise. We have to go to, there's the enhanced formulation, the spray dry dispersion, the HME, the lipids, and then there's the actual tablet and capsule. And And more importantly, what may even take more time in parallel with that is the analytical for that, because we have to be able to release test those capsules. And the test method, and that may sound straightforward, but if you hand a bottle of tablets to QC chemist and say, here, release test per these specs, well, what do I do? Do I grind these tablets up? Do I put them in a volley flask and dissolve them? If I do that, do I sonicate it? Do I mix it? What do I do? What exactly do I do? What filter do I use? And are the excipients going to interfere with the assay, the HPLC method that you gave me to begin with? So that work has to happen in parallel. And then the resulting test method, like the TME test method, the resulting test method has to be properly validated or phase appropriately validated. Now, get into phase one, that's relatively streamlined slabs. So specificity, linearity, accuracy, precision, LOQ, LOD. Those things are relatively straightforward, but they have to be done under GMP protocols. They have to be done under quality. They have to be reviewed by quality assurance. And a lot of times that analytical piece can take more time than the actual development manufacturing itself. So budget that into your timelines. And realistically, if you go to one of these more once we have that, we have to manufacture the product, we do the release testing, and then you're on into stability, and you can usually start dosing humans. Sometimes you may be able to. Some folks will go into first in human studies with preclinical stability, and they actually haven't got their GMP stability. But a lot of times we're looking. Customers say, "Well, I want at least 1 month of stability." So from the point we manufacture and do the release testing, you've got another month to get the 1 month stability on the back end. But the STD, HED capsules are 7 to 9 months. So we sometimes do a little faster. Micronized blending capsule would be 3 to 6 months. And then they're shorter than this, right? There may be micronized powder and bottle. What is that? It's just get the powder micronized, get it to the clinic, then before we start doing it. But what you may want to do, budget in somewhere along the way, is verifying that what is the right particle size, what is the right vehicle. So sometimes you'll have someone like ourselves or someone else do in vitro experiments to show suspendability, in-use stability, in-life stability, whatever you want to call it. So that doesn't necessarily free you up from doing some lab work, but it just takes a lot of that pressure off of you. Then cost. I'm not at liberty to say cost. We don't have CDAs, but and nor would I hazard a guess, because there can be these two different molecules can cost different things because there may be different iterations. But I will tell you, you're probably looking at least 2x between this and this. Whatever that is, double it. Take-home message: know that patient. Most important of all, know that molecule. Understand the phys chem. Understand the pharmacokinetics and to some degree the pharmacodynamics. Understand your clinical strategy, not just today, but tomorrow and the next day and the next day. And then again, you guys are entrepreneurs. You know your business strategy. Is it to hit that milestone and partner this thing, or is it to take this horse all the way? And that may go into your calculus on what you're willing to do on something, or what you need to do, what your investor, what your partners are telling you. Ask them what they want to see. That's what I would do. And then use this knowledge and plan ahead. I would say, look, we're always here to help you. We are a vendor. You pay for our hands, but we build a relationship by giving you our brains for free. And she mentioned we have a large scientific network, and one of the things we do as the S&T team is to be the connectivity between all that scientific network. So maybe you have a question or something down the road, say, hey, I want to know something, and I may not know the answer, but I have the ability to reach out to the 1,200 plus scientists that work around the globe for Catalent and say, have you ever seen this? Someone's asking about this, and I may be able to get some answers for you, pull papers for you, things like that. Again, we're just trying to build relationships with new customers down the road. So I think that's it for me. I'm gonna pause and take Q&A, and I hope that answered some questions for you, and maybe it created more questions, and which is probably a good thing. So anyway, thank you very much for your time. Appreciate it.
Questions?
Yes.
You have thousands of customers. You have breakdown the percentage of those are coming from academic world.
I think so. Yes, I can't get percentage wise. I'm trying to guess most of the customers was, to me, I'm not trying to be pat in the answer, but I'd say all of them is it all starts in academia, right? I mean, that's where a lot of it comes out of. So a lot of well, I guess that's not true. There are discovery companies that are working on that, but a lot of times you find when you start pulling it back a little bit, they're digging in academia for the research, right? So they're starting to go in there. So even when I'm talking to a customer we've known over time and we're a discovery company, they may be doing it in-house, but they may also be out there hunting themselves. So it's very common for us it's completely normal for me to go sit down with her with like we're gonna go visit a client, kind of hear what they have to say. We don't really quite know what they need yet. And we sit down, there's two people in the room. There's a CEO who may or may not be from the investment side, probably from the investment side, and they may or may not have a scientific background. And there's the CSO or the co-founder who's been working on this for years. And they're the ones that are really excited about it. They're the ones you really want to talk to because they're the ones that know the molecule. And they can tell you what you need to know on the back end. So that's a very, very common situation for us.
I think percentage wise, like if you looked at it percentage wise, big pharma versus small to mid-size, actually 84% of our business is small to mid-size companies versus the big pharma. I think sometimes people think, oh my gosh, Catalent, they're only going to work with the big pharma companies. Totally untrue. Because I mean, people coming into early phase programs, a lot of those big pharma companies, they do that piece in-house themselves and come to us later on. So the majority of our customers that we see coming in, our partners, are on the small to mid-size.
My group almost talks exclusively to small mid-size companies because, we're talking to a, big pharma X, they're
on the commercial side.
On the commercial side, but even we will sometimes get involved with discussions kind of just to facilitate, make sure that they're talking to the right people. But at the end of the day, they kind of, they know where they want to go.
Mm-hmm.
They've just, sometimes maybe they've probably done spray drying already in house and they're looking for a commodity. They're looking for who can come spray dry it. So they know they figured out what they want to do. They just, they're a little downstream from that first slide I had with all the early stage stuff,
I guess one of the approach to increase liver survival percentage after liver, the first test effect is people use this pro-jogger, right? Do you see those kind of?
Yeah, we see pro drugs all the time. Yeah. We actually, and so that changes the way you're looking at PK, right? I mean, it's not, we want to see the metabolism. That's what we're looking for. Sometimes people use pro drugs can also be used to improve permeability. And so we see that as well too. Drugs that are not orally available as the active moiety, but if you can do a pro drug, you may be able to improve the nature of that compound, look full nature, and get it to be absorbed. So I see that sometimes as well too. But yeah, go ahead.
What about some unusual small molecules? So we are in a space where our small molecules that we are designing right now, they're kind of like modular system. They have a pegylated backbone, for example. Then one is a pharmacophore, one is a targeting moiety, if you will, kilodalton, 1400. These will never be absorbed. And so, these are things that we were thinking about, and we probably don't need to give them continuously. It's really just like you give a few shots of that to start something. But they would be, IV formulations or maybe even sub-Q. Do you have some experience with those?
Yeah, absolutely. Yeah, we do. So it sounds like you're in targeted degraders. Sounds like you're describing a targeted degrader.
So these molecules will actually stay extracellular, but they will bind to a receptor, and then they will be internalized and then activated there.
Okay.
Yeah.
Hans, did you sign up for a one-on-one later? Or are you heading
So
out?
Shanghai is later because I actually have to fly back, and so Shanghai will do it virtually.
Yeah,
Yeah.
I think that's a.
Yeah.
I just want to reemphasize, I know we have to switch over. I hope everybody really took a look at those timelines. And this is part of why people get delayed is it's not too early because, as you know, we spent a lot of time talking about formulation and different modalities, but this is important as you're sort of now thinking about PK/PD, those kinds of things that can trigger molecule redesigns. And if you start working it out, if you're talking to investors and stuff, and they say when you're going to get to clinic, well, you have to at least have an idea of what your formulation is going to be in order to go do your tox studies in order to get to the clinic, right? So you start stacking that up and you work backwards from, your FDA submission for your IND. And you realize that this can hold you up, not just in 6 months, but it can push your timelines a year if you're not thinking about it early enough. So just really, really like to emphasize that, because you need to know, at least if you're gonna be up in that top level, 3 to 6 months versus the bottom level.
Or something shorter. Sometimes it's shorter,
Yeah.
sometimes it is.
Yeah.
Probably some people get away with it.
Some people get away.
And that's great. It's, you do, that's good.
You can get lucky and find out, just like all of us when we do our assignments last minute, you can find out that it's actually really easy, but you
Yeah.
can also find out it's not. And you
Yeah.
promise investors you're gonna
But
get
she
it.
left, but BD hates when I put the timelines up because, it's like our competitors are going to show something a lot shorter. I'm like, oh, okay, that's fine. It's fine. Yeah.
Yeah,
Yeah.
the shop that tells you the shortest timeline is not always the best.
Well, I just want to be realistic and, I highlight that, emphasize that when you go to a CMO and you do contract manufacturing, or you build your own plant, I don't care what you do, you're going to have to, there's the GMP piece of this, we have to be able to demonstrate we made what we said we made. And how do we do that? So that I'm dead serious when I say that analytical and validation piece takes as long as anything. So, so that's important.
All right. I'm gonna take a 5-minute break, come back at 10:40, and then we'll start on the next session.